I actually am not going to talk to you about gamma delta T cells, but about really a small little project. I'm a basic scientist, but I'm interested in asthma and in treatments of asthma and the associated pathologies. And we made a little discovery in mice, and we wanted to find out if the same that we found in mice holds also true in primates. So this is what our project is about. And now I have to figure out how this pointer works, and that looks good. And so I realized that in the title of my project there are a lot of terms that are probably not familiar to everybody. And so I will spend a moment trying to explain airway hyperresponsiveness and some of the other terms, then move on and tell you a little bit about our finding in mice, and then what we did to try and transfer that into primates. Now, what is airway hyperresponsiveness? It is a pathologically increased narrowing of the airways, and this happens when airway smooth muscle contracts more than it normally contracts in response to allergen exposure or also non-specific stimuli, for example, cholinergic agonists, even cold air can induce that. And other terms that describe the same thing are bronchial hyperresponsiveness, or you might have heard of twitchy airways. And I brought you these two images. Maybe we can just turn this down for a moment. This is MRI, but an unusual kind of MRI. It uses magnetized gas, and so you can see the air spaces in the lung very well. And this is what happens if methacholine is introduced. And now, all of a sudden, the air spaces shut down, and especially here you see it in the periphery. You don't see these light areas anymore. So this is the airway response, and hyperresponsiveness, this response is particularly strong. Now, when does airway hyperresponsiveness occur? It's associated with a number of diseases, including chronic and serious diseases such as chronic obstructive pulmonary disease or asthma. The exact mechanisms underlying airway hyperresponsiveness are still not fully understood, and they may vary to an extent with the diseases with which AHR is associated. But in allergic asthma, the immune response plays a large role, and in particular, lymphocytes that we call T lymphocytes are essential in the development and the regulation of airway hyperresponsiveness. And to study this mechanism, learn more about it, people typically turn to animal models, as we have as well. And so I want to show you a little bit of preliminary work in mice. And the collaborators in this study include several investigators, former investigators at National Jewish, and Irving Gelfand, who is still present in the show, and Rebecca O'Brien and myself. This is the cartoon that shows the model that we use most often. Mice are sensitized by intraperitoneal injection with an allergen, followed sometime later by an intranasal allergen challenge. And at this point, after they have been challenged, they develop a strong immune response and asthma-like features, which include airway inflammation and an allergen-specific T-cell response and airway hyperresponsiveness. The way we find out about airway hyperresponsiveness is we challenge them, again via the airways, with the cholinergic agonist methacholine. And then we assess lung function in these mice. Now in this mouse model, we found that the story becomes even more complicated. So different types of T cells have different effects on airway inflammation and airway hyperresponsiveness. Some mediate it, others just only enhance it when the mediators are present, and still others suppress this phenomenology. But all of them contribute to airway hyperresponsiveness. Now we can identify these different T cells by protein molecules they express on their surface. These protein molecules have also a totally different function. They serve as antigen receptors for these lymphocytes. And these molecules are known as T cell receptors. Because they're very interesting, they have to do with the specificity of the immune system and so, they have been studied a lot. And immunologists have generated specific antibodies against the T cell receptor molecules. These antibodies not only recognize the T cell receptor molecules, but they can also kill the T lymphocytes that express them. And if they can kill the T cells, then they can abrogate their functions. And so in our model, the antibodies against the T cell receptor can abrogate airway hyperresponsiveness. And the antibodies can be applied in various different ways. They can be injected. And we found that even inhaled anti-T cell receptor antibodies can be effective in preventing airway inflammation and airway hyperresponsiveness. So I want to just show you some results in the mouse model to document this. What you see here is a differential cell count of mononuclear cells in the airways. And I just want you to focus on this one, on these two columns here at the end. These are eosinophilic cells, which are key cells in airway inflammation. And this black column here designates the airway inflammation in an allergic mouse. And in a group of allergic mice, and another group which had the same inflammation was also treated with an inhaled anti-T cell receptor antibody, and the inflammation goes away. Not only are these antibodies effective against inflammation, but they also affect airway hyperresponsiveness. What you see here is the airway response to increasing doses of methacholine. And what we do to measure airway response is we measure a parameter called lung resistance. And this lower curve here represents the response of a normal animal, and the upper curve, the response of an allergic hyperreactive animal. And if we add anti-T cell receptor antibodies, again in the form of an aerosol that's inhaled, airway hyperresponsiveness goes down significantly. So the mice are less affected by their allergic status when they're treated with anti-T cell receptor antibodies. Now, these findings, from a scientific perspective, and that's why we started the study in the first place, provided direct evidence that T cells inside the lung mediate these pathologies. That's why we were interested in doing the experiments. And then once we had done them and gotten the results, we realized that these findings also suggest that anti-T cell receptor antibodies introduced in the lung might be actually used in the treatment of airway inflammation and airway hyperresponsiveness. And so for this reason, we were interested in seeing whether one could translate this result in rodents into primates, one step closer to humans. And so we set up a collaboration with the research group at the National Primate Research Center at UC Davis. They have the monkeys, and we identified an asthma model that they actually established and that seemed to be suitable for our purposes. In this model, adult rhesus monkeys are sensitized and challenged, in this case with actually a human allergen, house dust mite antigen, and they develop an allergic response. And eventually, some of the monkeys, after repeated challenges, will also develop airway hyperresponsiveness and the hyperreactivity to a non-specific stimulus methacholine. This model was published several years ago by one investigator in this group. And here you can see the experimental protocol. It is very complicated, but basically it consists of many, several sensitizations and many challenges of the monkeys with this allergen, house dust mite antigen, and eventually, when their pulmonary function is tested, some of the animals will be hyperreactive and behave like an allergic patient with asthma. Now, there are a number of challenges in translating from the mouse model to the rodent model. The rodent and primate lungs differ anatomically and physiologically. There are differences in the actual models that are used. For example, the mouse model is much more acute. We just sensitize briefly and challenge the monkeys. This model is more a model of a chronic disease, as would be the case in humans. There's enormous variability among the monkeys. They are kept sort of large cages, 200 monkeys at a time, where they sort of form little communities. They are outbred, and there's a lot of individual variation, not like the inbred mice. Because of the enormous cost of the monkeys, we can only afford very small experimental groups. And there are no antibodies against monkey T cells, so we have to use anti-human T cell antibodies that happen to cross-react with the monkey T cells, and so we have to make sure that they're actually cross-reactive. In our case, in the end, only one antibody was available to us that satisfied these criteria. It was an anti-human CD3 antibody. That's an antibody that will recognize all T cells across the board. And it did so with the monkeys. Now this is an abbreviated sketch of the study design. We had 6 female adult rhesus monkeys, 2 controls that were not sensitized, and 4 experimental animals, 2 of which did not during the various challenges become very hyperreactive, even so they were allergic, and 2 others that became hyperresponsive. What you can see here, this is basically the original model as established at UC Davis, and then we added for our experiment a treatment with a control antibody, a non-specific antibody, followed by pulmonary function tests and analysis of lung lymphocytic infiltrations and skin allergy, and then a treatment with a specific anti-T cell receptor antibody, that CD3 antibody I was talking about, and again followed by pulmonary function test. The question was, would this specific antibody have an effect on the hyperresponsiveness of the monkeys? Initial evaluation of the monkeys showed that, at least lucky for us, because we couldn't change the animals anymore, all of them reacted with the anti-CD3 antibody, and the treatments, both with experimental and control antibodies, were well tolerated. There was, as predicted, a high variability with regard to the initial allergic status of these animals. And in the experimental group, we had 2 animals that were only mildly responsive and 2 animals that were highly responsive, as I mentioned. And after the treatment, we determined that there was no significant effect of the antibody treatment on the allergic skin responses in our groups. There was also no clear effect of the antibody treatment on eosinophilic airway inflammation, although in one animal there was a strong effect. But there was a clear effect on airway hyperresponsiveness. And that I want to show you here. And what you see here are the 3 groups of animals. each two monkeys, 2 and 2. You can see that monkeys in the control group have sort of an intermediate, there's actually something missing here, I apologize. That must be the transfer to the different computer. What you see here is this scale is a scale that determines that shows the methacholine dose. So the higher up in the scale, the less sensitive an animal is, because it means a higher dose of methacholine was used. In pre-treatment, you can see the control animals have sort of an intermediate sensitivity. In the treated animals, there are these two that are not very sensitive at all. And then we have the two highly sensitive allergic animals. Now, post-treatment, these allergic animals are totally normalized in terms of their airway responsiveness to methacholine. And at this point, not all the data are in yet, and I just the functional data I showed you, I received yesterday evening. But what we can say so far is that in the monkeys, as in the mice, T cells in the lung contribute to the hypersensitive state, which leads to the uncontrolled responses to methacholine. And the treatment with the anti-TCR antibodies that we used via the airways causes clearly a desensitization and normalizes responsiveness to non-specific stimuli in the hypersensitive animals. The question is, and our study design didn't allow to test for that as well, whether the same treatment also has an effect on specific allergen responses in these animals. And I would like to acknowledge the support of those from the state of Colorado and from National Jewish Medical Research Center. Thank you very much.